Hello, I'm Denise from the Ensembl team, and I'm going to show you how to explore the region in detail view. I'm going to explain how to visualize genes, sequence variants, and other data along a genomic region. We will use the Ensembl Region in Detail view. The Region in Detail view is one of the most popular and widely used pages in our browser, due to the large number of data tracks that are available to display and compare. Let's explore the genomic region of the human lactase gene, or LCT, in this view. The LCT gene is associated with lactase persistence. This is the home page of Ensembl version 68. The Ensembl version changes every 2 months, and if it has been updated at the time you are watching this video, you are welcome to use our archive site. From the Ensembl homepage, search for Human LCT. Expand Gene, then click on Human. The result gives us 3 options to explore the LCT gene, with a gene, location, or variation focus. Let's click on the location link, which will take us to the Region in Detail view of Ensembl. This link tells us that the gene is located on chromosome 2, and these are the start and end coordinates. -1 means that the gene is on the reverse strand, and +1 would mean that the gene was on the forward strand. We are now in the Location tab, Region in Detail view. Let's explore the 3 images in this view. The first image at the top shows a schematic representation of human chromosome 2, with its banding pattern indicated. This red box shows the location of the LCT gene. You may also note some red and green bars across the human chromosome. For more information on haplotypes and patches in Ensembl, please have a look at our video, Patches and Haplotypes. The second image, in the middle of the page, shows a 1 megabase region, with genes displayed in the region 500,000 base pairs upstream and 500,000 base pairs downstream of the human LCT gene. The genome is displayed as a blue bar, and the alternating dark and light blue represent the different contigs, or blocks of sequence, that make up the whole genome assembly. We can see the neighboring genes on either side of LCT and their orientation. Those on the forward strand have a greater than symbol, whereas those on the reverse strand have a less than symbol. The genes annotated in this 1 megabase window are color-coded. Gold genes are protein coding and include manual annotation from the Havana project. Blue genes are processed and therefore non-coding. Purple blocks are RNA genes, and gray indicates pseudogenes. We can also identify on which chromosomal band LCT is located. It is on Q21-3, the long arm of chromosome 2. The third and final image in the Location tab zooms in on the LCT gene. All the Ensembl splice variants, or transcripts, are displayed. Boxes are exons and lines represent introns. Open boxes represent the untranslated regions, or UTRs, whereas filled boxes show the coding regions of the transcript. also known as the coding sequence or CDS. The transcripts are drawn underneath the blue bar, which means that they are on the reverse strand, indicated by the less than sign here. The human LCT gene has two transcripts in Ensembl. The gold one is protein coding, and the blue one is a process transcript that undergoes nonsense-mediated decay. Let's click on LCT 001. The pop-up window shows the transcript is a merged transcript with annotation from both Ensembl and Havana. Merged protein coding transcripts are colored in gold and are high quality as both projects agree on them. We can also see a transcript called CCDS 2178.1 in the CCDS track. This is a transcript belonging to the CCDS project, Consensus Coding Sequence, and its annotation has been agreed on between 4 different groups: Ensembl, Havana, NCBI, and UCSC. Note that LCT 001 corresponds to CCDS 2178.1 in its coding regions. We can zoom in and out a number of different ways in this page. With my mouse, I will draw a box around one exon and click Jump to Region. We could also move the bar on the right-hand side or click on plus to zoom in or minus to zoom out. Once we play around with the zooming options, we can always recenter the view on the human LCT gene just by clicking on the golden transcript and selecting the location link. You can also change the base pairs directly in the location box or recenter on a different gene. In the Region in Detail view, there is a set of data tracks that are already selected or turned on by default. In Release 68, these are chromosome bands, human cDNAs, RefSeq ENA, merged Ensembl and Havana genes, also known as the GenCode set, contigs, the CCDS set, regulatory features, sequences that may be involved in gene regulation, and percentage GC content. If we are not sure what some of these are, we can hover over the track name and then the eye or information icon. We can turn tracks off here at the track name or change the way they are displayed. Data tracks can be displayed in different styles, such as expanded with labels, among others. For a complete list with descriptions of track styles, have a look at this FAQ. Hover over the cogwheel for the human cDNA track. These are shown in a stacked format. Change it to normal. Now we can more easily see that there are 2 cDNA sequences that are shown on the genome above the blue bar. These are on the forward strand. and 2 that are shown below the blue bar. These are on the reverse strand. You might be able to see that the cDNAs in this region support the LCT 001 transcript. To make the comparison easier, click and drag the cDNA track next to the gene track. You can also click on a cDNA to find out what it is. These are just the default tracks. There is a large selection of other data tracks we could show. For example, there are 428 different data tracks for human to choose from in this release. How can we add tracks? Click on Configure this page or the cogwheel icon. First, we are taken to an Active Tracks window. These are the default selections. Note if a track is turned on, but we don't see any information drawn in the view, it means there is no data for that track in the region we are viewing. For example, there are no Ig T-cell receptor genes in the LCT region. Let's add some tracks. The available data for display is separated into this menu at the left. The main menu headings are Sequence and Assembly, for example, markers and clones, Genes and Transcripts, for example, different gene sets and RNA-seq models, mRNA and protein alignments. This shows sequences from public databases such as NCBI-RefSeq and UniProt aligned to the genome, ncRNA, non-coding RNA, other DNA alignments, variation, SNPs and small-scale insertions and deletions, along with larger sequence variation. Somatic mutations, short and larger scale variations from projects like Cosmic. Regulation, sequences potentially involved in gene regulation, for example, transcription factor binding sites, DNA methylation, and histone modifications. Comparative genomics, multi-species comparisons, oligoprobes, probe sets from microarray platforms, repeat regions, repeats determined with repeat masker, information and decorations, settings, display options, change the width of your view. Depending on which species you are looking at, you may have a subset of these menus available. If you have uploaded or attached data, there will also be a Your Data menu. Let's add tile path, clones, and markers. Under Sequence and Assembly, we have a few options such as markers and miscellaneous regions and clones. The 2 tracks we want to add are under those 2 options. There is 1 track only under markers. We will click on the link in the left-hand side menu and then turn on the track marker. If you click on the eye, you can get more information about the track. The star allows us to select a track as favorite. If we do so, it will appear in this menu. For the miscellaneous regions and clones options, there are 13 tracks available and none are displayed in the Region in Detail view by default. You can see the number here says 0/13, meaning 0 out of 13 possible tracks are on. Let's click on the link to see all the 13 different tracks and then select Tile Path. Click on the eye to find out what the tile path track means. These are the clones used to determine the genome sequence. What if we were unsure which menus to go to? We could have searched for marker or tile path by typing the term in the search box labeled Find a Track. We can type the first 3 letters of the search term to find the track. Typing MAR reveals different menus in which these 3 letters appear. We could have selected the marker track from these search results. Once we select tracks, save and close the menu by either clicking on the tick or anywhere outside the window. Have a look at the new tracks we just added. What if we want to draw the same marker track in the 1 Megabase view above? Click the cogwheel icon from the left-hand Configure this page button, as we did before. We can now navigate to the Configure Overview Image tab. The second way is to click on the cogwheel from the 1 Megabase region. We see a limited menu selection for this region. Select Marker as before and close the menu. Note that if we want to save this image as PDF or another format, click the picture icon to do so. Let's move to another gene to demonstrate some more track styles. I will click and drag my mouse to form a box around my new region of interest, the UBXN4 gene. I'll click Jump to Region and configure this page once again. Let's see what is in the RNA-seq models menu. I'm going to turn on brain intron spanning reads with variable height track style, and also the brain RNA-seq genes. With the expanded with labels option. We can now see a transcript structure based on RNA-seq evidence. It matches the exons in the CCDS sequence and in UBXN4001. This supports the hypothesis that UBXN4001 is expressed in brain tissue. The intron spanning reads look good, as they are stacked high. If we click on one of these blocks, we see the read coverage. For more about RNA-seq models, see this video. We can save this collection of tracks rather than turn them on one by one in a future session. To do so, click Configure this page, click Save As. Let's name these Markers, Clones, and RNA-seq Brain. Even if we now change the track selection, we won't lose our configuration. For example, let's turn off the RNA-seq tracks by hovering over the track name and clicking the X. We can go back to the configuration menu and load our saved configuration. Note, if you do register, which is free, and log in, your saved configurations will be available no matter which computer you are using. If you don't log in, favorite tracks and configurations are only saved to one browser session. Once a track is displayed, we may want to copy the URL link to send a colleague this region with the track turned on. We just need to hover over the track and then over the link icon. Right-click on this link. We can use this URL or send to colleagues so that this configuration can be seen by others. After all the changes made, we may want to go back to the original default set of tracks. We can do this by going back to Configure this page and clicking on Reset Configuration. Note, we could also reset the track order. Finally, one more tip. Depending on how it is displayed on your browser, you may want to change the width of the image in the Region in Detail view. Click on Configure this page, then on Display Options, and choose from a variety of dimensions or the best fit option. Save and close the window for the updated display parameters. That's all, folks. Thank you for watching, and if you have any questions, feel free to contact us.